Hello everyone. Today I'm going to talk about phagocytosis. So the learning objectives of today's classes, the definition of phagocytosis, what are the steps involved in phagocytosis, and what are the mechanisms of destroying the microorganisms, that is the killing mechanisms, and the applied aspects uh, regarding phagocytosis. So coming on to phagocytosis. Phagocytosis is the process of ingestion and killing of microbes or other foreign substances by phagocytic cell. Basically, phagocytic cell is a leukocyte. So, leukocyte, we know it is classified into granulocyte and agranulocyte. In granulocyte, we have neutrophil, eosinophil, and basophil. And agranulocyte, we have monocyte and lymphocytes. So, mostly all the cells are involved in phagocytosis, but the predominant cells which are involved in phagocytosis are neutrophil and monocyte, and one more cell which is involved in phagocytosis, macrophages. So the coming on to the steps involved in phagocytosis, as the slide tells, that is margination is the first step, diapetes, chemotaxis, opsonization, and engulfing or ingestion of microorganism and killing of the microorganism. So these are the 6 steps involved in phagocytosis. So you will discuss about each and every step. First step is the margination. So margination is actually the leukocytes are attracted to the site of the injury. So this is the site of injury actually, and after that injury there will be inflammation in the site. So what happens next? The leukocytes are attracted towards the site of injury. So from the blood vessels. Okay, so these are all the blood vessels, mostly the capillary. This is the capillary endothelium, and which is adjacent to the site of the inflammation. So what happens? The WBCs are line up adjacent to the endothelium, and in the endothelium there are substances called chemicals called selectants. Okay, the selectants attract and attaches with the uh, WBCs, and they are lined up in the endothelium. That is called margination, and they move along the margins of the endothelial cells, and they find the opening in the endothelial cell to enter into the site of the injury. So this is what is called as margination. The next step is the diapetesis. Diapetesis is nothing but the entry of the WBCs into the tissue space from the capillaries. From the capillaries, they enter into the site of the injury, that is to the interstitial space, into the tissues. So that entry is called diapetesis. So there are pores in between the endothelial cells. Okay, so the WBCs adjust their shape. They adjust their shape, and they enter into the pores into the tissues. So this process is called diapetesis. The next step is chemotaxis. So the chemotaxis is actually the movement of the WBCs from the blood to the site of the injury. Okay, so after the diapetesis, the cells move into the site of the inflammation. So the process by which leukocytes are attracted towards the foreign pathogen or at the site of the inflammation is called chemotaxis. So the bacterial products interact with the plasma factors and cells. They produce chemokinins. So the substances are called chemokinins. These chemokinins attract the neutrophils and other leukocytes towards the infected area or inflammatory area. So these inflammatory markers are called chemokinins. The predominant most common chemokinins are complement proteins C3 and C5a, and also leukotrienes and cytokines. All these are collectively called as chemokines. So basically, when the bacteria interact with the plasma factors, okay, plasma factors, they produces these inflammatory markers. So these markers, they attract the WBCs towards them. So the WBCs moves towards the site of the inflammation. So that is called chemotaxis. The next one is the opsonization. So opsonization is actually again the plasma factors like opsonin. They coat the bacteria to make them tastier for the phagocytes. So what are the opsonin materials? They are IgG, immunoglobulin G, perforins, and again the complement protein C5A also act as a opson. So basically what they do, this is the bacteria. These are all the complement and immunoglobulins and perforins. They coat surrounding the wall of the foreign antigen, then makes the foreign antigen tastier for the phagocytes. So they neutralize the negative charge in the surface of the bacteria. So the main function of the opsonin is they neutralize the negative charge so that uh, they cannot be repelled by the phagocyte membrane. Okay, so by neutralizing the negative charge, they are attracted towards the phagocytes. So phagocyte easily attaches with the bacteria. So that is opsonin. Okay. So next stage is the engulfing or ingestion. This is actually the stage of entering the foreign pathogen or bacteria into the phagocyte. Okay, so this is called engulfing. Okay, the foreign pathogen with the help of pseudopodia and form phagocytic vacuoles inside it. So you can see that in this diagram there are two pseudopodias arising from the phagocyte. So they surround the foreign bacteria or pathogen, and then there will be internalization of the foreign pathogen, and the pathogen enters into the cell inside the cell of the phagocyte, and they form a phagocytic vacuole. It is the phagocytic vacuole. And after that, what happens? The lysosomes and the granules present in the cytoplasm of the phagocyte attaches with the vacuole and form the phagosome. Okay, the lysosome granules of leukocyte fuse with the phagocytic vacuole, and then they form the phagosome. Okay, the phagosome is nothing but it has both bacterial substances and also the fused vesicles of the lysosome and granules. The next stage is the killing or uh, uh, the destruction of the foreign pathogen. Okay, so as I already mentioned, the the killing happens by two mechanisms. One is non-oxidative killing, another one is oxidative killing. Okay, so the non-oxidative killing is actually by the lysosomal enzymes and the granules, enzymes in the granules and all these things. Okay, so the lysosomes and granules, they contain bactericidal agents like degradative enzymes, proteases, and defensins. So by degranulation, they kill the bacteria through disrupting their outer membrane and break the DNA. So what happens? So this is the green color is the bacterium. So once the bacterium is entering into the cell, it forms a phagocytic vacuole. So this is the lysosome, which is de- uh, uh, which degranulates into the phagocytic vacuole and forms the phagosome. And after the degranulation, so all the substances, that is degrading enzymes, proteases, defensins, they act on the bacteria. They breaks the bacterial membrane and breaks the bacterial DNA, and then they kill the bacteria. Okay, so finally the waste products are formed inside the cell, and they get exocytosis out of the cell. So this type of killing is called non-oxidative killing. The other form of killing is oxidative killing. Again, when the neutrophil and phagocyte gets activated, okay, it also activates an enzyme called NADPH oxidase. Okay, that enzyme produces reactive oxygen species which are antimicrobial in nature. Reactive oxygen species which are antimicrobial in nature. So during this process, what happens? The oxygen uptake also increases. Okay, during the process of oxidative killing, or when the phagocyte is getting activated, the oxygen intake by the phagocyte is also increased. So that is called as respiratory burst. Respiratory burst. So when the oxygen intake is increased, there will be formation of superoxide anion and free radicals are formed inside the phagocyte. Okay, we call it as oxidants or free radicals. So these are superoxide anion, hydrogen peroxide, hypochlorous acid, and singlet oxygen. So these uh, free radicals or oxidants are produced by the action of the NADPH oxidase and also one more enzyme called as superoxide dismutase. So this is superoxide dismutase forms the hydrogen peroxide, and the other enzyme is called myeloperoxidase. This myeloperoxidase forms the hypochlorous acid. So all this produced inside the phagocyte. All these are free radicals. They are oxidants. Uh, they ultimately they kill the bacteria. So this is what we call it as oxidative killing or oxidative burst or respiratory burst. So this is how the phagocytosis happens. Okay. So now coming on to some of the applied aspects of phagocytosis. So one is neutrophil hypomotility. Okay, a disorder, a primary phagocytic dysfunction of neutrophils, which decreases the phagocytic activity. And the next disease is chronic granulomatous disease, which occurs due to the defective NADPH oxidase. So the NADPH oxidase is defective, then the free radical formation will not be there, and the oxidative killing will not be happening. Okay, so that is one of the diseases. It's called chronic granulomatous disease. The another one is amyotrophic lateral sclerosis that occurs due to the defective catalase enzyme. Catalase is also one of the enzymes present in the phagocyte. The main function of catalase is it converts free radicals into uh, water and oxygen. So basically, catalase is an antioxidant. Basically, catalase is an antioxidant which converts excessive hydrogen peroxide into water and oxygen. So in persons with the defective catalase function, what happens? It is a genetic disorder also. So what happens if the catalase is not there or defective? The excess hydrogen peroxide will not be converted into water and oxygen. So the hydrogen peroxide is accumulates inside the cell, uh, which causes destruction of the cell itself. Mostly it is present in the spinal cord. Okay, mostly it is present in the motor neurons of the spinal cord. So it affects the motor neuron. So it is a type of motor neuron disease, which we call it as amyotrophic lateral sclerosis. Okay, with this, I conclude the phagocytosis. I hope you all enjoyed and you all understood the mechanism of phagocytosis. Thank you.